So welcome everyone to this last event in our 2025 Spring NASA SITSI Leaders Series. I'm Sarah Kirn, the Participatory Science Strategist for NASA out of the Gulf of Maine Research Institute in Portland, Maine. And I produced this series with Mark Kushner, NASA's Citizen Science Officer, Brianna Putnam and Jennifer Shirk of the Association for Advancing Participatory Sciences, and Heather Fisher of Crowd Informatics. And as always, today's program has been shaped by their ideas and insights. I may be the mouthpiece, but I am not the brains, and I could not do this without my wonderful colleagues. So thank you all. Today, we have the distinct pleasure of learning about the Open Science Data Repositories Analysis Working Groups, the latest addition to the ranks of NASA-supported citizen science projects. I want to warmly welcome everyone involved with the AWGs into this NASA citizen science community. I hope that today's event and conversation helps all of us, newcomers and the old guard alike, see ourselves as part of an innovative, open, and inclusive community of science enthusiasts and scientists. I'm still wrapping my head around the work of this remarkable group of volunteers who include everyone from high school students to research scientists and data scientists around the world. If you, like me, are new to space biology, hang on to your seat. This is going to be extra fun for us. What I was quicker to see and what I'm really excited to talk with you about today is that the OSDR AWGs, as we call them, are another example of a citizen or participatory science project that invites volunteers into the full process of science and even into the driver's seat. Let me back up and give that idea some context. I suspect that there may be some folks with us today who are not yet familiar with citizen science or how NASA defines citizen science. So we'll start there. A note about the name first. There are a lot of names that are in current use to refer to scientific work done by collaborative groups of people that may or may not include trained scientists working within their fields. If you are part of our regular citizen science conversations, I invite you to put your favorite term and your definition of it in the chat so we can get a flavor of all the terms that are in use. Many of these names, I bet all the definitions won't change. The names are kind of evolving. Many of them are beginning to refer to more discrete practices or applications of these methods. We're going to talk more about that later. But for now, I'll just say that citizen science and participatory science are the terms most commonly used as umbrella terms for this work. Citizen science is also the term most commonly seen in the research literature about these methods and applications. NASA defines citizen science as simply science projects that rely on volunteers. If you want the specifics, and I encourage you to check them out, look at NASA's 2-page science policy document on citizen science, which we affectionately call SPD 33. So I'm going to talk about 3 of the big ideas in that document. The first one is that citizen science projects are on the hook for producing new scientific knowledge, just like any other research supported by NASA. They're held to the same rigorous standards. It's how many publications of new research does a project produce in peer-reviewed literature? Second big idea, citizen science projects need to provide volunteers with a meaningful experience. And this one is kind of a self-fulfilling or self-determining idea, right? Because these are volunteer projects. If you're not giving your volunteers a quality experience that's informative and meaningful and satisfying to them, they're not going to stick around with you long enough to produce good science. The final big idea I want to talk about is this idea that these projects are open to anyone. Now, note that this does not mean that all projects have to be easy enough for anyone to do, but rather that anyone who wants to participate could, if they have or they develop the required knowledge or get the required equipment or can get to themselves to the place if there's a geographic element in order to participate. It's perfectly okay. I think it's even appropriate to have some projects in our portfolio that require significant prior knowledge or developed knowledge or equipment or skills. That's fine. In a typical NASA-supported project, the project leaders, who are usually scientists, have posed a research question that requires a collaborative effort among humans to answer. So there are two common examples of projects. The first one I'll talk about is projects that need observations to be made, collected, and compiled from a broad geographic area, or even around the globe. A science team can't be everywhere at once at the right time, so they need to engage a distributed network of observers. Many of the NASA-sponsored Earth projects and some of the heliophysics projects like Aurora Source and Spectacular that need observations made from Earth follow this model. Another class of projects or examples comes from research that depends on human eyes and brains to classify large data sets, like those produced by space telescopes or other space-traveling remote sensing instruments. Machine learning and artificial intelligence can help to winnow down those data sets, but as of today, humans are still an essential part of this kind of research. All the Zooniverse-hosted astrophysics and planetary science projects fall into that category, and there are lots of others as well. So both of those models are what I would call a task-focused or a research question-focused project. Participants are primarily, not exclusively, but primarily there to do a defined task. The Open Science Data Repository Analysis Working Groups are a really different kind of project. In this model, we see a group of people, lots of them on the call with us today, coming together to work collaboratively in science, not around a single research question, but around a scientific resource, specifically the Open Science Data Repository that Brian's going to tell us about in a few minutes. One really cool thing about this model is that it puts participants, volunteers, in a context where they're invited into the full scientific process, from framing the questions, preparing the data, conducting literature reviews, or doing the data analysis and writing up the results. It might not be expected or necessary for every individual participant to do all of these activities, but it's not unusual or outside the regular task of the project for participants to pose and pursue new research questions, or even propose new analysis methods. It might be a stretch goal, but it's not extraordinary the way it might be in a task-oriented project. Now, it's important to point out that the OSDR AWGs are not the first projects that we've seen where volunteers are invited into the whole scientific process. We have some great existing examples. The Ham Radio Science Citizen Investigations, or HAM-SCI, and Radio Jove project are two projects that are each focused around a scientific tool, ham radios and radio telescopes, respectively. And those projects each have an open invitation to participants to propose and pursue research using those tools. We also have some great examples of task-oriented projects, and here I'm thinking about Disk Detective, the Backyard Worlds projects, and a few others that have created a space where advanced users and scientists can come together in real time, virtually, to talk about the science, the data, and open up new avenues of research. And that's an incomplete list. Those are just some examples. I still think it's fair to say that as a field and community, this citizen science practitioner community, we know a lot more about how to engage, motivate, and reward volunteers in projects focused on a question or task than we do on recruiting and supporting volunteers and working with a scientific instrument or a data repository in an open-ended context. I'm really excited to connect those projects together to learn from one another, but also to help the rest of us expand our thinking. So that's why I'm so excited for us all to take a peek under the hood of the Open Science Data Repository Analysis Working Groups today to see what fresh ideas and common challenges we might find. So let me introduce today's speaker. Ryan Scott is the science lead for the Ames Life Science Data Archive, which merged with the Gene Lab data system to form that Open Science Data Repository in 2021. Ryan is now the liaison between the Open Science Data Repository and the analysis working groups. And having seen Ryan in action at the recent AWG and Catalyst Summit, I recognize him as a fellow champion of collaborative, participatory citizen science and a champion of people, like so many of you in our audience, who come together to give their time, energy, ideas, and talents to advancing scientific knowledge. It is an honor to welcome all of you, Ryan, and all the members of the OSDR AWGs we have with us to our community and to this conversation. Ryan, over to you.
Okay, I believe it's sharing full screen and pretty. Thank you so much, Sarah. Yeah, it looks like I saw a head nod, so I think that's correct.
Looks good.
Wonderful. So, hi, I'm Ryan, and I'm today speaking in my capacity mainly as the point of contact liaison for the OSDR analysis working groups. And yeah, what an honor to be here. And I just see so many familiar names in the chat. I hope I do y'all proud for how much passion and love and energy and just like exceptionalism that the analysis working groups. And what a huge honor to be brought into the fold with NASA Citizen Science in general. And I'll explain a little bit in a little bit on how that evolution happened. So it's interesting you mentioned that, Sarah. I want to first start with biology and sciences on Earth before we talk about life sciences and biology and health in space. I think that's probably the place to start because so many people who are maybe new to space life sciences might not also be familiar with biological and health sciences on Earth. So the place of data and data sharing and accessibility to that data has really changed the place of repositories and access. And that's why the Open Science Data Repository, which, as you mentioned, Sarah, was the merging of OSDR and GeneLab and a couple other things, is so important. And there's just been a renaissance. Like, this is a tweet that will always stay in my brain, seared. Francois Chollet was at Google. He invented Keras, which is a deep learning library. And he said within 10 to 20 years, nearly every branch of science will be, for all intents and purposes, a branch of computer sciences, right? Computational physics, computational biology, computational archaeology, realistic simulations, ML everywhere. And I think about that tweet so much from 2021. And it's just, I mentioned it, and I point at that because of how computational sciences and data sharing across on Earth has changed. That is, cancer researchers share data and do computational research. Same thing for genetics research, chemistry, the whole field of chemistry, molecular biologists, clinical data, and as well as, and so like these are all just examples of like different portals across those domains. And I think data sharing and access to that data sharing is so important on a number of levels. Reproducibility, transparency of results, to support public policy, to return data to the people that created it, to give credit. Visibility into negative, quote unquote, results, also failed clinical trials, speeds up results and reporting so that some of these studies don't take years and years, that data gets back quickly. Also education, the access for future generations to learn from the results on all the data. Of course, today, one of the big things the AWGs is involved in is getting this data to enable new science, the reuse of data for that, as well as the number of different breakthroughs and ventures that can be had once data is released. People can invent stuff, they can develop platforms. It's amazing. And arguably one of the most important things on data sharing, in my opinion, is also to increase the trust of science, the science process, and the believability. So I give all that because that sort of sets the stage for where the field is on Earth. Now let's think about how, because we're living in the beginning of the second space age. This is from a Nature release from last June that shows just like this tremendous increase in the number of objects that have launched into space in the last few years. Like when I started at Ames in 2014, little did I know that I was joining at a time that would lead to this tremendous, I mean, that's at least a magnitude of order, whatever. But it's just this tremendous increase in the time that we're living in. And to help urge and push humanity to become multiplanetary, to help life thrive in deep space, there are a number of health and biological hazards that we need to figure out the basic science for, the applied science, even the various operational outcomes to help make sure and enable health to be sustained and ecosystems to be sustained in space. And so the hazards is crucial. The big two are microgravity and radiation, but here are a number of others that need to be considered. And all these hazards to ecosystems, biology, and health to boot, some of them interact, right? So what happens when you have impacts to your heart from microgravity, but you also have ionizing radiation to your heart, and maybe you also have these weird microbial impacts to your cardiovascular system. All of a sudden, single little impacts become a combination of multiple stressors. So I thought it's important if you haven't really thought about this field of space biology, which is different than astrobiology. Astrobiology is the search for and detection of life across the universe, the search and detection of life across the universe, whereas space biology is really to help life thrive in space, to help push humanity to become multiplanetary. And that's why I want to mention the Biological and Physical Sciences Division of the NASA Science Mission Directorate, for which these working groups, the Open Science Data Repository and Gene Lab, it's the primary stakeholder, very proudly part of BPS. And here's just a snapshot of how BPS sees these various different experimental platforms in Earth, in low Earth orbit, to lunar orbit, to Mars, the various different locations where science can and will be done and will continue to be done for a number of different purposes. And I'm also part of the and OSDR and the AWGs are also supported by the NASA under BPS, the NASA Space Biology Program, which sort of helps to determine the basic science, the fundamental life processes and mechanistic processes, genetic or otherwise, that sort of have these types of ecosystems in spaceflight, so that we have the basic understanding of how life adapts and responds to space, and also can then help develop applications that can help counteract the ill effects of spaceflight. And NASA Space Biology has been around for decades, in effect, right? This is the graph's not too important, but it shows you historically from 1975 to up to 2016, the series of publications that have been consistent over the decades and a real wealth of knowledge garnered from them. And so I want to come back to the data question. Data unto itself it's interesting to think about maybe 15, 20 years ago versus now. Making data reusable is going to be essential to help life thrive in space. For knowledge discovery, data is going to be essential to help enable the various different knowledge graph, machine learning, computational systems biology approaches. Data is going to be crucial to understand the impact. So that is it truly microgravity or is it truly radiation, to narrow out the different independent variables and the factors. Data is going to be crucial to enable analysis across missions. So you can look at, well, what happened in a mission in 2015 versus 2020? Were they the same duration? Was it the same species? Was it the same conditions? That type of thing. And so it's just a list of the various different reasons that data is going to be so important. Data is also going to be crucial to help develop these new platforms, right? There's a number of different commercial payloads and other international space agencies and NASA, of course, that are involved in developing the lunar and future Martian ecosystems and health support platforms and countermeasures that are going to be crucial for life to thrive in space. And I think reusable data is essential. And I think the AWGs are a perfect example to involve the entire world in spaceflight. It's probably my favorite sentence to say out loud, but how wonderful it is that instead of just watching the next moon landing, when science is delivered to the next moon landing or Martian mission, you're not just going to be able to watch it. You're going to be able to hopefully have access to the data and actually be involved to analyze that data and contribute. Like, what a tremendous change from, let's say, 1971 versus now, right? So this helps involve the world in spaceflight. And there's some links that I'm sure a colleague will share in the chat connected to some publications if you're interested more on those types of topics. And I credit to David Van Valen at Caltech that originated this analogy. But I also want us to think about the place of data. And the reason we're talking about all this data is because that's what the AWGs are investigating, feasting upon, and analyzing, and finding all that stuff. And I mentioned the Tycho Brahe era of space biology and space health, et cetera, because most people probably might remember from high school or college physics about Kepler and his laws of celestial mechanics. But probably less people are familiar with his mentor, Tycho Brahe, who spent his entire life developing methodology for astronomy and how to analyze the stars. This is pre-telescope. He was really the last great pre-telescope astronomer. But he had spent great detail in developing some systems to collect data. And I mentioned that because he collected and curated and annotated data that then Kepler was able to data mine, in effect. I'm not sure Kepler ever called it data mining, but I like to think about that example in terms of like last year's Nobel Prize in chemistry that DeepMind had on the protein folding question using with some of the collaborators with Google DeepMind. Well, just like Kepler was enabled by the work that Tycho Brahe had done, so too was this Nobel Prize on DeepMind on protein folding, developed upon decades of data collection, curation, and annotation, and hundreds and thousands of students, I'm sure, PhDs, that provided data to these various resources, which then the Google DeepMind and their collaborative teams worked together on the protein folding challenge. So I mention all this because that's how I see OSDR and the working groups, right? That OSDR has spent an inordinate amount of time on data standards, data processing, structures, metadata, as much as possible to every different aspect of any factor that could impact the science to collect, curate, and annotate that data. And this also has led to a great number of different discoveries, new basic fundamental science, be that renal, kidney health, to skin, the integumentary system, to cardiovascular, to plant, to mitochondria, to bacteria, also helping divide and develop applied in countermeasures, right? So mining this data to help develop and invent new countermeasures for spaceflight. And data has been mined also from OSDR that helps give mission planners and operational support the types of things that are needed to build habitats and safe and healthy ecosystems of the future for lunar and Martian missions. And the breadth and volume of data is important. So when you think of like one study, let's say you're studying microbes on the ISS and the microbes that were a specific class and whatever. It's one single mission, one single organism. Maybe you do like an imaging and maybe you do a genetic analysis, but and that's one paper and that's amazing, but it is a limited view, right? What's beautiful is what happens when you bring these studies together. Right? Let's say the microbes were there at the time that the humans were on the station, that there were also plants, and you were investigating, I don't know, cardiovascular or skin or something. When you begin to look at studies together and looking across species and across data sets, across missions, you start to get more context, right? And in many different data types. It's not just genetics. It's not just microscopy. It's not just, swabs to measure like the microbial content. All of a sudden, when you start putting this all together, you're getting a full, complete puzzle of what's happening in space. And really the thing that enables this is quality data, number one, but also the volume and breadth and reusability of the data. And reusability is just so essential. And then you can do meta-analysis, systems biology, and all of this is contributing to having better scientific readiness for crewed missions to extend humanity to become multiplanetary. So, so like Sarah mentioned, Ames Life Sciences Data Archive, NASA Gene Lab, and also the biorepository called the NASA Biological Institutional Scientific Collection merged in the last few years and formed OSDR. And across OSDR, there's lots of different data types. I think a tremendous amount of data volume for being such a niche repository. There's also different APIs, which I'll mention a little bit later, for better data access. If you're new and don't know how to do bioinformatics, or let's say machine learning, we also have on-demand trainings to help increase your skill set. There's also the data is also findable, accessible, interoperable, and reusable. And we also have these series of tools that as well, and tutorials, which another colleague will probably put in the chat on the tutorials if you want to learn more. This is just a bit of a snapshot. I'm going to go through these sort of more quickly. There's something like 103 different data assay types, meaning is it a microscope or do you do a sequencer? Those would be two different assays. And so in the last several years, I think we're up to 103 different assays that we've spent time standardizing the data, the metadata, the process level of the data. It's sort of tremendous. And that's a lot of where the AWG began, actually. I'll talk about in this slide or two. And there's been a tremendous return on investment. right? You can sort of think of it, there's like 109-ish publications of data that have submitted their data to OSDR. And now we have these 537 studies, 118, actually, if you include student theses and preprints, it's really closer to 150 publications have been enabled by OSDR ever since GeneLab started as the real nexus of this concept a number of years ago. A number of people here probably have never really thought about biology and space necessarily. So I do have this interesting slide that sort of tells a story from an ISS mission that had rodents on the space station, right? You had rodents on this, it's a rodent habitat. And after the mission or during the mission, there were a series of dissections of the ex vivo rodents. And this led to a number of tissues that were deposited in a freezer in Ames, the biorepository. A collaborator, actually not a working group member, AWG member up in Canada, made a tissue request to the NBISC, right? He then published about this muscle study looking at calcium uptake to the muscles, as well as looking at proteins using Western blot of those muscles from Rodent Research 1. I think he also did Rodent Research 9. Anyhow, that investigator, how great is that? They had a publication based off these tissue requests, fantastic. And it contributed new knowledge that had never been seen before. What's amazing is that this person that made the tissue request then went and submitted its data to OSDR, ALSDA, and GeneLab. And there were a bunch of other studies that were conducted with these rodents that submitted the data to ALSDA and GeneLab. But here, and then this just sort of shows the life cycle. A collaboration which involved the AWG in 2022, 2023, mined a lot of this data, right, across Rodent Research 1 and Rodent Research 9. And they then mined the data and used this explainable AI tool called QLattice to try to understand at the molecular level what's driving the physiological stuff that you can see in a microscope level, what's genetically driving the physiology. And so what a great life cycle to show the return on investment, which otherwise would not have been possible for this new knowledge from because this machine learning explainable AI paper also had new proteins and tried to identify, which is perfect for countermeasures. And then you can go and validate these results in the wet lab. So it's a beautiful revolving circle. This is just a snapshot of some of the data that's available in OSDR, spanning a number of different tissues, spanning from rodents to Drosophila to microbes, you name it. There's 537 studies. I also want to go a little bit quickly, but when I talked about the data standards that enable the reusability of this data, it has to do with metadata, which is really about the data, right, on top of, or about meta, I don't know if that's Latin or Greek, but the metadata, right? So this is like the study design. What was the duration? What was the species? Was there a strain of the species? What were the preservation techniques? That type of information. The data itself, if anyone has ever tried to crunch data before, you might know that spreadsheets can be terribly painful. And so the other thing that we do with a lot of the tabular data is we transform them to make them easily and simply readable, as well as clearly delineated on their nomenclature and their units. And there's a great deal and a beautiful collaboration with especially our working group on pipelines for sequencing data. So these are standard agreed upon consensus pipelines that our working group has collaborated with us to develop. Because, and I'll show you in a slide, but these types of processing pipelines are critical to enable more people to be able to analyze the data. And lastly, for great access options, there's a number of different APIs. For example, just recently we released the new OSDR Biodata API, which is a great way to help bring data locally to your own computer instead of going one by data set in the browser. And all these data standards have enabled a tremendous ability to not even just help, let's say, a bioinformatician run analysis, but now you can develop platform tools which make it extremely straightforward for anyone, even with just basic access to the internet, can visualize omics data. They can go look at the environmental telemetry from these missions. So that's O2, CO2, radiation, relative temperature, and also purely on radiation, because there's a number of different biophysics and radiation dosimeter questions that a number of people, so you can actually compare shuttle versus ISS versus lunar versus heliocentric radiation data in RadLab. So Oh, and this is just a bit of a snapshot of, like, for example, last year there was a tremendous amount of publications that came from OSDR. I think almost 20 in 2024 were purely from the analysis working group, which is tremendous. That so many publications came just from the AWG. This is a snapshot across the different types of publications that have come from data being mined. Lots of methodology and tool development, which you'd expect to help develop the standards for the field, but also stemming from plants to microbes to muscle to immune system to central nervous system, cardiovascular. And I was sort of proud to see an analysis of, yeah, of course, there's a large percentage of the authors of people that have mined data are across North America, but it expands much more broadly to show that there is opportunities across the whole world to just, it's all open access data. And this is also a last snapshot about across the types of research disciplines that a lot of these publications have come from, of people, including the working group, that have mined these data sets. So like I promised before, to give you a bit of the history of where that working groups are at, right? So I started with OSDR, but now to really finish the rest of my talk just on the AWGs, because you sort of have to have that setup point about, okay, this is the wealth of data, the types of data, the standards of data, which then can, feed this community to access data. But at the very, very beginning, when GeneLab began, it took a lot of skills. We're talking PhD level, computer science bioinformatician levels, and it was tremendously, and it's all raw data, right? And so taking raw data, we then, that the need for the AWG was born, because it's like, can we take raw data and develop these consensus pipeline standards? Because instead of on the level of terabytes, once you have these process levels of omics, genomic data, right, you then only have gigabyte level, which makes it lowers the bar so that many more people can analyze and look at that data. But on top of that, to get to those visualization tools, now it's even one step further. Okay, now that we have all these process data, now we can develop these visualization tools that anyone with a computer with basic following of the prompts in the visualization tools can do analysis. And this is really where the AWG began to grow. And I said, okay, now that we've standardized all this data, let's now reuse this data. Why not? It's all open. And so the AWGs began to grow, and anyone was welcome to join. Yeah, there was still a core of the higher level academic expert, core facility type people, but it started to grow, and it sort of said anyone is welcome to come and join and mine this data together. And then to boot, once the Ames Life Sciences Data Archive came and said, well, what about the environmental telemetry data and the phenotypic imaging data and all the other data types? Now you start having this whole new level that's not just on the molecular omics level, but it's like, what about all of the life science relevant data? And then, of course, the renaissance in machine learning and computational approaches last year, it sort of just kicked the door open even wider to say anyone is welcome to join, and we have this tremendous growth of the AWGs. So welcome to the analysis working groups, right? There are 9 of them currently. You'll see Amber, Afshin, Daniela, Elia, Kirill, Begum. I'm the ALSDA AWG chair, Lauren, Richard, and these are the 9 AWG chairs. So if you're new to the working group, it's really important to contact those. If you're in animal AWG, make sure you contact Amber. If you're part of the human AWG, make sure you contact Elia, because they run monthly meetings. And so what is the AWG? It really has 2 major purposes, right? So these working groups really accomplish 2 things. One is they provide feedback on data standards, be that data of data coming in or data leaving the repository to make it more reusable. And the other thing is the working groups obviously collaborate on data mining and investigations. And you can see a number, and I'm sure a colleague will put in the chat. You can go look at the over 100 publications that have been enabled by GeneLab and OSDR. And there's just been this tremendous growth. In 2023, we had about 300. What is it? I think it's upper 300. About at the end of 2023, we had just under 400 people in the analysis working groups. And as of last week, yeah, I think we're actually closer to 1,200 now, technically. And here's the geographic distribution, which is amazing, right? As you would expect, USA has a good majority, but it is open, and we try to make our meeting times as conscious as possible. A lot of times, if you're in the Pacific, it's very early Pacific morning to be conscious of colleagues in India or Japan or Europe or elsewhere. Yeah, there's just been this tremendous growth of the AWGs. And we had to figure out a way to scale this up, right? 300 to now 1,000 plus people. And so we were playing around with different ideas, and we really needed to have control of the system. So we developed what we call the AWG forum space last year. Actually, this morning we crossed 1,000 people are now in the forum, the AWG forum space. And so this is a snapshot of the About page. And the reason we developed this is because we kept hearing from AWG members, you need to have better transparency so that everyone knows about all the groups, that this isn't any buddy system. Like we added clear authorship guidelines in the charter. We need to have better communication, because across that many people, emails were becoming wild, because it was hard to keep which AWG with which group, so many emails. And just the ability to organize, right? We're talking 9 AWGs, 25 subgroups. It's quite something else. And so with the forum space, right, it's like a Reddit for space life sciences now, right? You can post interesting articles that are relevant to space life sciences. There's opportunities, maybe there's funding, there's little notices on meeting updates, and there's also a group member hub. And here's all 9 of the group member hubs, because it's one place where it's one-stop shop to go and check out, okay, I want to find all the recordings from the ALS AWG meetings, and I want to look at find the link for the running notes and the minutes. And what's that link? I don't have access to my Outlook today, and I'm on the go and I'm driving, but I'd like to listen in. And so that you have the meeting links there. And to boot, there's the calendar, right? And we do our best to keep this as accurate and up to date as possible. But as you can imagine, there's a lot of changes, and depending on people's schedule, things have to move around or things get changed. And there's just an example of like in the AI/ML AWG, here's the DNA imaging subgroup calendar invite. So, and this is a snapshot of all those subgroup projects. I know it can be overwhelming. And if you're new to the AWG, I would take your time. You need to sort of learn, and I would focus. Like I notice a lot of times when people join AWG, they join 7 of them. That's cool that you can join all 7 AWGs, but I would suggest you consider choosing maybe a topic that you think you might be strongest in or find the most interesting, and then really sort of, commit towards one AWG and one project. And as you begin to get the hang of things, then maybe you consider, or maybe another thing, or start a new one, right? I mean, all of these AWG groups started because someone said, hey, I've got an idea. Maybe that'll work. Maybe it won't, but maybe it will, right? That's how all of these different groups started. So if you have an idea and you've been mining the OSDR data and looking at it and say, I think there could be some interesting evidence on the thymus. I've always thought about metabolism in the thymus and the mitochondria. What hasn't then been analyzed? Well, that's an interesting point. Let's bring it to one of the AWGs, contact one of the leads, right? Contact one of the leads, and maybe consider presenting that idea at one of the future meetings. All right. So, and if this is all completely brand new and you would like to learn more skills, I'm going to highlight 2 open access, free, on-demand, asynchronous courses. One is called Gene Lab for Colleges and Universities. It was built in a college environment. That's where a lot of the curriculum was trained and built at first, but now it became completely open access and on demand, which is amazing. And it sort of steps you through the introduction to this is space biology, this is some basic coding skills, and this is some basic understanding of how to read sequencing and genetics. And then it brings you to more advanced concepts to help do analysis. And the other one is this TOPS T training. So we won a grant a few years ago from the SMD. That is an AI/ML curriculum that James Castelletto and Lauren Sanders developed. And this also too is open access and free and on demand, asynchronous. And it also just it involves the curriculum. There's recorded lectures. There's Google Colab as well as scikit-learn aspects to it to give you materials to actually not just learn, but actually test yourself and actually have some back and forth learning on how to do and use machine learning for spaceflight life sciences. And if you're interested in the AWGs, this is the join form. I'm sure a colleague will probably put it in the chat. We do try to onboard people as soon as possible, but things can get tricky. And so I will at least say every couple to few weeks, I wish we could do it more often, but every couple to few weeks, we also, I don't know if it's a good idea to really automate that, because a lot of this is built on relationships and actually asking questions and meeting people where they're at and having ideas. And and when you join, I would urge you to look at the charter and the code of conduct. There's my contact information if you're interested. And so, just to end this, I do want to say everyone is welcome. We've never turned away one single person from joining the working groups. We've had high school students be on collaborative AWG publications, different people that might be more interested in having ideas on creative outreach type stuff. It's not necessarily like, there's also been the citizen science literature reviews, right? Maybe there was a physician somewhere around the world, and they were like, "Listen, I don't have a lot of internet access. I would just love to touch space sciences and space biology and learn more." And so them and 100 other citizen scientists began analyzing publications and mining data. So, and just to end that, I think thousands can engage in making life multiplanetary through open science and citizen science, which is great. So, and yeah, all are welcome. So I really appreciate everyone's time, and of course, sincerest thanks to leadership at NASA Space Biology, Biological Physical Sciences Division, SMD, and NASA Human Research Program, and the passion and love that our team itself, the OSDR team, to create these standards in the database to provide this data to the community. I can't tell you just what an exceptional team and how proud I am of everyone on it. So thanks. And I guess we got a great deal of Q&A now, so that's perfect.
Oh, thank you so much, Ryan. Wow. There's so much. I kind of feel like we should have listed 9 different new to NASA citizen science community projects, like one for each of your analysis working groups. They cover such a large kind of scope of biological sciences, and then of course in space. Yeah, so thank you so much for that introduction. That was great. And you gave us some hints about how you are working to organize. You as one person are working with this community of 1,036 people to support and enable them. And really, I mean, obviously you're not organizing everything. They're really doing it all. Can you share a little bit about your kind of what's your role in this community? What do you spend your time on? And how have you kind of kept this group being so productive? What do they need from you?
So I do think it's important to give credit to the AWG chairs of each AWG themselves, because that is largely where a lot of the activity is. I think my role predominantly is to facilitate bringing people in and asking questions. Or several months ago, there was a young person, I think they were just starting college and they're new to biology. That's fine. And they were asking questions like, I don't know what I'm interested in. And so on the forum space, right, we actually have an app that you can just have on your phone, which is amazing. And you just sort of have a chat like, well, what are you interested in? And, maybe after the next AWG meeting, let's stick around for 5 minutes and we can talk about your ideas. And then eventually I'm like, well, I'd love to learn machine learning. And I was like, oh boy, do I have an option for you? And so then it's just like, yeah, this is basic. And this learning is a lifetime endeavor, right? So it's like picking up skills here, picking up skills there, maybe contributing to a paper. I mean, there's nothing better than when someone contributes. I mean, we have a running list of hard to quantify success metrics. Someone that met someone through an AWG meeting that then they went and submitted a grant together and they won it, right? Or I was at a meeting a couple of years ago, a young person from, she was in India, tapped me on the shoulder. I never met her in person. And she's like, she was so excited and I hadn't met her before. She's like, I'm so-and-so. And I'm like, oh my gosh. She said, I can't tell you how grateful I am. I was working at 10 p.m., 11 p.m. at night. And everyone thought, why are you spending all this time volunteering with NASA? And now I just applied and I got accepted to a university in the US and I got everything settled and I'm starting my PhD, and none of this would have happened if I hadn't stumbled upon this years ago. And I'm part of my first manuscript, and that's amazing. So there's like all these little success stories that are hard to quantify per se, that are just like, it was almost emotional when we had, because I wish we could do more in person like, where's that great? Oh, perfect. I think I'm sharing my whole screen, so I think I can just do this. And this was such a good write up, but I can't tell you how important this meeting was in
Yeah,
person, and because we've been collaborating through COVID together, right? Obviously, and 90% of us had, even people on the OSDR team had never met, right? So when we were able to meet, it was just perfect, and it really became just this sort of I've always thought of this as citizen science, right? It became this sort of like
good.
stalwarts of citizen scientists sort of placed across the field,
Yeah,
that would bring new people in, and it ebbs and flows, right? Someone else is like, hey, I just got this new position and I'm going to have to pull back. Do I have to stop being an AWG member? I'm like, no, just stay in touch, keep tabs, and it people ebb and flow with life and family, and there's complications with work and everything. So
yeah,
the activity, it definitely it goes through waves. So
great.
yeah.
Yeah. So I know, speaking for Mark and myself formally, we're so excited to have you in the NASA Citizen Science portfolio of projects and to hear about the productivity of your community. Of course, it just gives us so much more to be proud about. So we're thrilled to have you here. And as I said in my intro, I'm really excited to learn more about how, like, these are in some ways kind of self-organized groups of mixes of professional and volunteer scientists, would you say? Like professionals working in their field, I should specify. Can you clarify or share a little bit of the range of what's the balance in most of the working groups or in a subgroup working on a project between scientists working in their field and people who are just passionate about the topic and interested in learning and contributing?
It's hard to pull true demographic because there's tricky things for us to ask a lot of those types of questions per se, but
Understood.
at the same time, it's sort of this little bit dependent on what the project is. Like there was the literature mining exercise, not exercise, the project, which is still ongoing, the literature mining for cardiovascular data, which recruited, it actually reached out to SEDS, the Space Exploration DS, something students. Oh, exploration student. It reached out to these two international global student organizations, put a call out like, hey, are you interested in reading Space Life Sciences paper and contributing to the Citizen Science Project? Join. And so we had 80 people, not just young people, I would stress. These were true citizen science across the gamut. And they met and they got training and they had a peer review system to validate each other's results. And like, for example, that was, I would say, 80% true citizen science. I mean, I wouldn't even call them, I mean, words become weird on classifiers. It's like you have these
Yeah.
people, but we also brought in, okay, now that we have all these results, I'd like to recruit 10 cardiovascular SMEs and to make sense of it. And it's a great learning experience, especially as they're interpreting the results. People are like, well, we saw this. What does that mean? Is this the best way to represent these results? And so but other times it might be skewed the other way, right? It might be 80% professionals in their field, and it's more 20% maybe younger or non-professionals that are just want to be involved. Maybe they've learned a skill. Maybe they found a machine learning model, and they're like, hey, I'm getting into coding, and I'm just figuring out how to do this, and I think I can complete these analyses. There's someone else I could maybe pair up with and ask him questions. And so other collaborations are more, it's a bit of a mix, especially across that many, with 20
Yeah.
20 25 projects. It sort of spans the gamut.
Right. Yeah, and one of the bits of knowledge we have about citizen science projects, and again, I'm thinking more of those kind of more common formats of projects. The average volunteer is doing one project is doing multiple projects. So that kind of has helped us all think about how people move among projects as their interests and their skills and their knowledge change and grow. And there's a comment in the chat from a Katerina about this is the new way of people finding groups of colleagues to work with. And this really does feel like the vision of open science kind of coming to fruition. And of course, it's going to be a continuum. And I would love to see us work to to figure out more pathways from your very complicated, challenging, like kind of one-off projects in some cases, with some of the projects that are more just, frankly, they're fun. Like, it's really cool to look at galaxies nobody's ever seen before, and build up the vocabulary and the models and the movement around this community. I think it's just super, super exciting.
I yeah, this is definitely a two-way street, Sarah. We're
I hope
interested
so.
in learning more, and some of the most exciting things, like, it wasn't an AWG project, but last summer, Heliophysics and RadLab were actually able to collaborate across life sciences data, radiation data that was on the BioSentinel mission. And they then took heliophysics data and lunar reconnaissance orbiter, whatever had the dosimeter on the moon. And they took data and they had a challenge to come up with, okay, is there a way we can do, space radiation impact predictions using machine learning? And I was like, how cool, we're finally, because we are like just a couple months ago in, it's not this DAG and radiation with Bayesian network. That's a astronomer that has a Bayesian network expertise and is interested in Rad Lab, and also the Human Systems Risk Board has a series of these what they call directed acyclic graphs. It's a long story, but my point is that also crossed because he was bringing tools from a completely different domain and saying, hey, I think if you guys really have this radiation data that's open in RadLab and you have these directed acyclic graphs, I think I might be able to, we could have a project that cuts across domains on, which is tricky right now. At the same time, there's also one of the most, this is one of my favorite things, when these AWGs begin to interact more, right? For example, the plant one here, the plant and human leaky gut nutritional analysis, I think that started in the plant group. They then asked the ALSDA group, is like, hey, do you know or anything about the gut data that might be had in OSDR? Or maybe can we also cross rodents and humans, maybe looking at the blood and gut data markers? And so it became this interesting plant human physiological genomic collaboration that if we didn't have this AWG, I don't think, because then a gut, gastrointestinal, subject matter researcher was able to join that group, and just these types of interactions that are just otherwise, like, I don't see how a lot of this would have ever happened otherwise. Like, it just ideas that would never have, and that's one thing I'm for sure of, is that there's someone else out there that knows more than I do on all, and any of us, right? But to bring in those new ideas.
And just to break in there, you said you hope it's a two-way conversation. It's not a two-way conversation. It's like a 40-way conversation. We have 32 live projects right now on the site, and you and I are not the only people who know stuff about this. So I'd love to open up to more questions. Ryan, thank you so much for this introduction to this incredible, generative, lively space of people and ideas and really cutting-edge science. It's really exciting to have you all with us. Thanks.
Absolutely. My sincere honor each and every day to be part of this. It's what we've poured our hearts and souls into this, and the community, I feel, is reciprocating, and it's a wonderful, growing, profound thing. So thank you.